How you doing? Right, I'm jet lagged to heck and back. So I've been away for a couple of weeks, and I've got a few slides here. We're going to look at the testis. After this week, it's going to be Christmas. So I'm going to be away for another couple of weeks, but I think if I remember, we'll go back to the ovaries. But the testis, I've got 3 slides here. I haven't looked at them yet, so I'm not entirely sure. We'll pick the best one, basically. But I always find that whenever I see the testis under the microscope, it's just instantly recognizable, doesn't look like anything else. So let's have a look at the testis under the microscope. We'll take our time. We'll start from a nice wide zoomed out thing, and then we'll look at higher power magnifications, and we'll talk about the different cells, and we'll see what's going on. But you'll see what I mean. Instantly recognizable. These are going to be sections of animal testis. I like the look of this one. This one is darkly staining, and it's ovoid shaped, so I guess that's probably the whole testis, which would be good, right? So if this is good, this is the only slide we'll need to look at. See, I'm guessing it's testis because look, see how it's got a... They're lettered, so students don't know what they are. Let's see if I know what it is. There you go. What do you think? Does that look like any other tissue in the body? Okay, yeah, nice. So if I go to the edge there, I can see a nice connective tissue covering. So on the outside, that's a very strong line, and it's nicely stained running all the way around there. That's the tunica albuginea, the connective tissue surrounding the testis that holds it all together and gives it support, and that connective tissue will go into the testis to give it support as septa, which will divide the testis up into lobules, but that's all by the by, really. And then, oh look, that's some fat up there, but deep to the tunica albuginea, there's a good example there, is the tunica vasculosa, the vascular layer. And I'm preempting myself a little bit. Let's just zoom in a touch. And there we can see a blood vessel. Oh yeah, this looks like a very nicely stained section. So the solid layer is the tunica albuginea, and then we can see a blood vessel that happens to have been caught longitudinally. We can see the blood cells inside it. So that's the tunica vasculosa, the vascular layer deep to the tunica albuginea. Hey, this is great. I've gone off topic. I'm already talking in more detail than I originally intended to go into, but if I don't know, if I see it, it's very difficult to not say it. Right, so zoom back out again. So this is my lowest power. This is a 4 times objective lens and 10 times magnification to my eyes, giving me a 40 times magnification. It'll be a little bit different to you depending upon the size of your screen. But so what we see then, if we look at the testis, sure, we see that tunica albuginea on the outside, but if we, if we look around, this is what the testis looks like. It's lots of circles. And the reason it's lots of circles is because these are the seminiferous tubules. The testis is made up of tiny microscopic tubules all coiled up and packed into the space of the testis, because like we've seen elsewhere in the body, we're aiming at maximum surface area. So inside, so these are tubes cut in transverse section, right? So this is a huge length of tube, And in that tube, in the edge of the tube, we're seeing a special epithelium, we're seeing germ cells, and those germ cells are going to become spermatozoa, get released into the center of the tube, get moved along the tube, and they're going to mature somewhere else, and then they'll get released if needed for fertilization, right? So we're maximizing the surface area by getting as much tube as possible packed into the testis to give as many germ cells and produce as many spermatozoa to increase the chances of fertilization, right? Fertilization, reproduction are massive drivers of evolutionary changes. So that's what we're seeing there. So these tubes are the seminiferous tubules, and they will be the focus of what we're looking at today. So we can see that some of those seminiferous tubules have been caught at a glancing blow, and that's why we're seeing different shapes there. All right, let's go back over here. These looked pretty good, right? That one in the middle there. And let's zoom in a little bit. So, again, I've also got off topic, and I haven't really organized myself very well, because we would always talk about the function first. I think people know the functions of the testis. The functions of the testis are to produce spermatozoa, the male gamete for sexual reproduction, for fertilization, and also there are cells in here making testosterone, and testosterone is a driver of male secondary sexual characteristics, as well as other bits and bobs like, you know, muscle mass and so on. So the cells here are becoming spermatozoa, helping those cells become spermatozoa, or making testosterone in a nutshell. Okay, so what have we got here? Well, it's another epithelium. So if we handle the spermatozoa side of things first, around the, so you've got it's a seminiferous tubule, so around the edge of the seminiferous tubule there's a basement membrane supporting the epithelium. The cells around the edge there, that's where we find the germ cells. These are the spermatogonia. And we're looking at the darkly stained nuclei. So the spermatogonia, the germ cells, will constantly produce more spermatozoa throughout life. They'll always be producing more spermatozoa, which is why old men can still be fertile. The spermatogonia around the edge, once they start to develop, so those cells are going to go through meiosis. They're going to go, it's to do with the number of chromosomes that are going to be formed. So they're going to go from a diploid number of chromosomes to a haploid arrangement of chromosomes. Oh, so easy to get into too much detail here. Looking at the cells, you've got the spermatogonia around the outside. Those spermatogonia are going to go through the process of spermatogenesis to produce spermatozoa. And they're going to work their way towards the lumen, towards the middle of that tube during spermatogenesis. And they're going to become so they're going to go from being spermatogonia on the outside to in the middle of the cells of the epithelium. They're going to go through stages of being primary spermatocytes and secondary spermatocytes. And then as they get towards the inner edge, they will become spermatids, and then they will become spermatozoa, and then they will be released into the membrane. So let's see if we can see that at the higher powers. Man, this is such a cool section. So you can see, I mean, you can quite clearly see there how you've got those spermatozoa with their flagella, their tails, right? This is, yeah. Don't know if this goes to my higher power or not. Right, so there we're looking at the epithelium, and then around the outer edge you can see the basement membrane forming the oval of the seminiferous tubule that has been cut through, and you can see darkly staining nuclei around, well, cells with darkly staining nuclei around the outer edge. Those are the spermatogonia. And you can see other cells then that as we move towards the lumen, the center of the tube, but still in those layers of cells of the epithelium, you can see there are cells with large nuclei, not a lot of cytoplasm. The nuclei are still fairly darkly staining. In fact, I think the other one's a better example actually, if I slide up. Sorry to my future self in editing. We can see the spermatogonia around the outside, and then we can see large darkly staining nuclei. Those are the primary and secondary spermatocytes, so they're going through meiosis there. And as we get close to the lumen, we can see that cell shape changes quite markedly to become spermatids. And then as we get right up close to the lumen, those spermatids have become spermatozoa. And the other cells in here are the Sertoli cells, or the sustentacular cells, or the nurse cells, and those are supporting the process of spermatogenesis. So you must have Sertoli cells for these spermatogonia to become spermatozoa, so they're necessary for fertilization. Okay, let's go to a higher power. Let's see if I can get a decent image. So in terms of Sertoli cells, so the cells that have got a small darkly staining nucleus, those are the Sertoli cells. And then the rounder cells that are darkly staining are the spermatogonia. And then as we move away from the basement membrane towards the lumen, we're seeing the spermatocytes. And as we get closer to the lumen, we're starting to see the spermatids and the spermatozoa. So after meiosis has occurred, a lot of the cytoplasm gets removed and the cell reshaped, and that's what we're seeing in the spermatids. All right, let's find a nice bit. Okay. What we're looking at here in this germinal epithelium is, sure, we're looking at the stages of spermatogenesis, but we're looking at cell division. We're looking at mitosis and meiosis. So how do you feel? Are we going to try and tackle cell division and link it to what we can see here? All right, so if we look next to the basement membrane, the outer edge of the seminiferous tubule, we can see those spermatogonia. And some of those spermatogonia cells are going to divide. Now, most of the cells in the body have got 2 copies of every gene. Genes are stored as DNA. That DNA is wrapped up and stored as chromosomes. So most cells in the body have 2 sets of chromosomes, which means they are diploid cells. They get called diploid cells. You might see the notation 2N. So if they've got 2 sets of chromosomes, they're diploid cells. Most of the cells in the body have this, and these spermatogonia cells have that. Now, the cells and tissues here have been stained with a couple of stains, and the purple stain is staining the DNA. So the dark purple that we can see there is DNA. So where the stain is darker, we've got more DNA clumped together, and when that purple stain is a little bit more spaced out, that DNA is a little bit spaced out. So we are literally looking at the DNA there. Now, the spermatogonia are a pool of germ cells, right? They're a pool of stem cells. So one of the things they're going to do is the spermatogonium will divide using mitosis, the normal form of cell division that most of the cells in the body do. Mitosis is great for wound repair and growth and that sort of thing. So the spermatogonium will divide using mitosis. And what it's going to do is it'll produce two cells, and one of those cells will become another spermatogonium, so that you've always got your pool of stem cells to make new spermatozoa from, and the other cell will become a primary spermatocyte. Now, if the cell is going to divide itself and make two new cells, it has to perfectly copy all of its DNA. So it has to duplicate its DNA and then put the same DNA in each of the two cells, so it's identical. So if you look at the spermatogonium and then we move to the next layer of cells, the primary spermatocyte cells, how do they look different? Well, if we look at the dark purple stain, we're just looking at the DNA, the nuclear material. But the cell is bigger than that. We can see the edges of some of the cells there, so we can see the cytoplasm of the primary spermatocyte. But if you look at the purple, the DNA of the primary spermatocyte, what do you think? Is there more DNA in the primary spermatocyte than in the spermatogonium? Yes, there is. Well, how has that come about? Well, when cells of the body go through the cell cycle, when they're getting ready to divide, they duplicate all their DNA. They're still diploid, because that describes them having two sets of chromosomes, but they've copied their DNA ready to divide into two new cells. And that's what we're seeing in the primary spermatocytes there. We are seeing more DNA. That's why they look bigger and darker, because there's more DNA in there, there's more stain. So they've doubled the amount of their DNA ready to enter meiosis, which is the other form of cell division, and the special form of cell division that we're seeing here, which is associated with sexual reproduction. Now, meiosis has two cell divisions. So the primary spermatocyte is diploid. It has two sets of chromosomes, but the primary spermatocyte is going to divide. This is meiosis 1, and that gives us the next cell, the secondary spermatocyte. So if you look at those secondary spermatocytes, we can again see that that dark purple is staining DNA, and we've got nice round circles, and that dark staining, that dark round circle, that's just us looking at the DNA, looking at the nucleus. If we look carefully, we can see the cytoplasm around the nucleus, and that's the rest of the secondary spermatocyte. Now, does it look like there's less DNA in the secondary spermatocyte than in the primary spermatocyte? Yes, it does. And that is what has happened, is that in meiosis 1, the secondary spermatocytes have now only got one set of chromosomes each. But they've kind of got... Talking about the amount of DNA and chromosomes gets very confusing very quickly. I blame the way chromosomes and the words chromosomes and chromatids get used anyway. So the secondary spermatocytes, these are the cells that are formed after meiosis 1, after the first meiosis cell division from the primary spermatocytes. And that DNA that was in the primary spermatocytes has now split between the two secondary spermatocytes that have formed as a result. So there's less DNA in there. We can see that under the microscope, and these are now haploid cells because they only have one set of chromosomes. But they've got enough DNA in them to divide again to each make two more cells. If I talk more, I'm going to make it worse. So meiosis 2, the second stage of meiosis, is where those secondary spermatocytes will then divide again, and each secondary spermatocyte will give two spermatids. So if we look at the spermatids, well, they look quite different again. So again, that dark purple is just staining DNA. And now, it's difficult to tell if there's less DNA or the same amount of DNA, because it's in it's squashed together. It's in a much smaller space, isn't it? So the DNA content is now being packed together tightly as they're going to become spermatocytes, and they're going to become this, or they're going to become spermatozoa and become this very efficient cell that will make its way through the female reproductive tract. So what's happened there? Well, the secondary spermatocyte is a haploid cell. It only has one set of chromosomes, but it has enough DNA to put one set of chromosomes into each spermatid. I know that sounds very counterintuitive when I describe it like that. If I ever find a straightforward way of explaining this super accurately, you'll be the first to know. But the secondary spermatocytes, each secondary spermatocyte becomes two spermatids. Each spermatid is also haploid. It has one set of chromosomes, and those chromosomes are then getting tightly packed together, and the extra parts of the cell are getting removed because they're not going to be needed. And then those spermatids will become spermatozoa. By the way, the name of the process by which spermatids become spermatozoa is called spermatogenesis. Now, why are we doing all of this? Well, we need to make one male cell with one set of chromosomes and one female cell with one set of chromosomes, and then we bring those two cells together and we make a new genetically unique individual with two sets of chromosomes, which takes us back to the idea I started with, is that we have two copies of every gene in every cell of the body. So we're creating a special cell that has one set of chromosomes so it can meet another cell with one set of chromosomes from the opposite sex, combine them and make a new diploid individual. So we get a haploid cell plus a haploid cell, put them together, fertilization, then creates a diploid cell. The other reason we're doing this is in meiosis 1, the genes get moved around, they all get mixed up a little bit. So the big advantage of sexual reproduction is the diversity of genes, mixing up the genes and getting different genes from two different parents. This can give big advantages in survivability and adaptation. I'm talking about evolution. So meiosis 1 is also the process by which genes get moved around and mixed up. So each spermatozoa can contain the same genes but in different forms. The more I talk, the worse I make it. But yeah, sexual reproduction gives major advantages to adaptation and evolution. That's why this is all happening. And then actually, we can see if we move around, so if we now know that the purple is DNA, and if you've ever looked at the stages of meiosis and mitosis in a textbook, cell division, and you've seen the DNA getting moved around in the cell as the cell gets divided, we can actually see that happening in some of these cells. Yeah, look, in those cells in there, so in the secondary spermatocytes, you can see the very dark purple DNA getting pulled to either side of the cell, just as we see happening in the textbooks. So the DNA gets pulled to either side of the cell, each is a copy, and then the cell gets split down the middle and pop, you've got two new cells. So yeah, we can see cell division in action. Cool, huh? You see what I mean though? It doesn't look like any other tissue. It's cool to look at. Oh, that's quite good too. So there, close to the lumen, we can see what was a much larger cell. It's quite small now. So those little dots those little purple dots close to the lumen, those are the cells becoming the spermatids. Now that's really good. So they've gone from being big dots to being little dots, much more compacted, right? Yeah. And the same there. So from the outer edge towards the lumen, we're seeing cells passing through spermatogenesis. Okay, so if Sertoli cells are the nurse cells, the cells supporting spermatogenesis, the other cells, the cells that are producing testosterone, are the Leydig cells, and they're also known as the interstitial cells. Interstitial. So that means they're in the tissues in between the tubules. They're not within the tubules. So look, there's a seminiferous tubule, there's a seminiferous tubule, there's a seminiferous tubule, and in between the seminiferous tubules there, that's our interstitial space, that's our interstitial tissue, and we can see blood vessels in there, we can see some blood cells. So the cells in here, these interstitial cells, these are the Leydig cells, and these are the cells that are producing testosterone. The other cells you're going to find in these spaces will be fibroblasts, which are producing the connective tissues, which will be helping hold all this together, right? So that's all the Leydig cells are. The interstitial cells, they're in those spaces there. They're not within the tube, they're outside the tube. Here's a good one. Bit of a glancing blow, but to the left there, you can see spermatogonia. You can actually see two different types of spermatogonia because there's a cell there as I go up and down through the focal plane. There's a cell up against the basement membrane which has a dark nucleolus, and there are other cells which are just darker generally. Their nuclear material is a bit more spread out. Does that make sense? So those are the spermatogonia. What I'm trying to say is not all spermatogonia look the same. You've got type A and type B spermatogonia, and they look a little bit different. And then as we move away from the basement membrane and we move towards the lumen of the seminiferous tubule, we start to see lots of circles. We start to see primary and secondary spermatocytes, and we can see that nestled up amongst them, there's at least one Sertoli cell there that is taller. It's not round, it's a taller cell, and it's got a nucleolus in the middle there, whereas the other cells all around it are round and kind of darker, and those are the spermatocytes. So the Sertoli cell is helping those cells through the process of spermatogenesis. Then as we move to the right even further again, as we get closer to the lumen, we can see how those cells have become much smaller. They're much smaller, darker patches of nuclear material. So the cell has become condensed, the cells are becoming spermatids. And then as we go to the right again, that nuclear material has become even more compacted, and they're becoming late-stage spermatids and then spermatozoa. And we can see those spermatozoa in the lumen. We can see those long strands of their flagellae all mixed together. It's the Sertoli cells that I think are trickiest to see. I think it's good to see this stuff in situ, isn't it? Because if you're studying spermatogenesis and whatnot, you look at lots of illustrations and drawings in textbooks, but if you can see the actual microscopic cellular anatomy in situ, I think that really sells it. Even if it can be a little bit harder to identify stuff that's not actually labelled. Right. And Christmas is here, so I've got a couple of weeks off again. Well, I've been working the last couple of weeks, but then I've got a couple of weeks off to recover, and we'll get back to this in a few weeks' time then, in the new year. But thank you for sitting with me, and There are so many inappropriate things I could say right now. Thank you for sitting with me and looking at the microscopic anatomy of the testis and better understanding the human body. All right, I'll see you in a few weeks.